my senior research project explores social complexity and brain evolution in an ultra-social species of ant. So humans are arguably the most socially complex species because of our ability to both form and maintain complex social networks, and we also have disproportionately large brains for our body size. In the 1990s, Robin Dunbar showed that neocortex size in primates is directly correlated with their social group size. So he proposed the social brain hypothesis, which states that larger brains are associated with greater social complexity. Since the social brain hypothesis, there's been a lot of debate about whether it holds up across primates and other social taxa, such as social insects. So in ants, social complexity is defined by an extensive division of labor, or splitting up of jobs amongst specialist workers. In my species of interest, Atacephalotes, there's an extreme division of labor. So they're therefore considered to be extremely socially complex. And they live in colonies of up to 10 million workers, so huge societies. Not only is there a division of labor between fertile queens and all of her sterile daughters, but there's also a division of labor among the sterile workers into what we call subcastes. These sterile workers range in head size from 0.5 millimeter to 4.5 millimeters, and they're behaviorally categorized into subcastes based on their size and labor roles within the colony. The smallest workers, which are known as minims, remain primarily in the nest where they take care of the fungus, the queen, and the larvae. The medium-sized workers are the ones that actually leave the nest, go out into the environment, and forage for leaves, and cut them and bring them back to the colony. And finally, the largest workers are known as soldiers. They have large, impressive mandibles, which make them well-suited to defend the colony. Basically, all of these different workers come together to form one superorganism. So based on the differential labor roles across the subcastes, these workers face different cognitive challenges within their lives. So basically, I wanted to explore how social life has influenced the evolution of brain structure and how brain structure influences behavior. So in order to quantify brain structure, I dissected 10 brains from each ATA subcaste and stained them using immunohistochemistry for a protein called synapsin, which is located in all the synapses of the brain. I then imaged these brains using a confocal microscope and then labeled them in a mirror to figure out what each size of the brain regions are. Next, I wanted to investigate how variation in brain structure affects the metabolic costs across the workers. I did this by measuring oxygen consumption rates. Basically, the more oxygen the brain consumes, the more active it is and the more calories it needs. Finally, I was looking at gene expression differences underlying the differences in brain phenotype. So basically, we have to look at RNA rather than DNA because all of the workers are sisters and therefore they share 75% of their DNA. So only the RNA can account for their subcaste differences. To do this, we extracted mRNA from whole brains from 3 different size groups, representing the minims, media, and majors. So first, I'm going to show you the patterns we found in brain investment across workers. So first, we looked at the optic lobes. These are involved in processing visual input. And as you can see from this graph, the smallest workers actually show the smallest relative investment in this brain region, which makes a lot of sense because these are the workers that work underground in the fungus, which is in complete darkness. So wouldn't it make sense for them to invest a lot of tissue in visual plasticity areas? We next looked at the antennal lobes, which are responsible for processing olfactory input. And it's important for you guys to know that ants rely on pheromonal communication in order to function in their everyday lives. So the antennal lobes are important for all the subcastes. But we see that there's a trend where there is greater investment in the antennal lobes in the 1.8 and 2.4 millimeter subcastes. And this might make sense because these are the workers that are actually going out and interacting with the environment, and therefore they need to process more pheromonal and chemical cues. Next, we looked at the mushroom bodies. These are large and very interesting structures in the ant brain, and they're implicated in learning and memory. We actually saw a very surprising result here, which showed that the medium-sized workers had the highest relative investment in this area. So we came up with the hypothesis that these medium-sized workers are likely task generalists within the colony compared to the minors and the majors, meaning that they have a larger behavioral repertoire and do more tasks than the smallest minims and the largest majors. If this is true, then they do need more tissue in the mushroom bodies because they need learning and memory qualities to be elevated in order to maintain their behavioral flexibility. Finally, we looked at the central complex. This is an area involved in sensory integration and motor control. It's homologous to the human cerebellum. And we see here that the trend is that the smallest workers have the largest relative investment in this area. This makes sense because they are trying to navigate through the fungus, which is like a maze, and they're also doing it in complete darkness. So they need very fine movements to like navigate through that. So I'm next going to go through the gene expression results, and we hypothesize that the variation we just saw in relative brain size is going to be reflected in differentially expressed genes across the subcastes. So this first graph represents the differences between the samples, and what's important for you to notice is there's very few differences between the minims and the majors, but there's a lot of differences between the minims and the medium-sized workers, as well as between the majors and the medium-sized workers. This graph highlights the similarities between the individuals, and once again, you can see that there's a lot of genes shared between the minims and the majors than either of those with the medium-sized workers. So my explanation for this trend is once again that the media workers in ADA are task generalists, unlike the specialist minim and majors, and therefore they have a different brain gene expression profile to help them maintain this behavioral flexibility. So we next scaled their oxygen consumption rates in order to evaluate the metabolic costs of these brains. So this graph shows a very predictable result, which is that larger brains use more oxygen, which makes sense because there's more tissue. However, what's important to notice in this figure is the extreme variation in oxygen consumption rates in the 1.8 millimeter subcaste, which once again supports our hypothesis that these are medias because some tasks like leaf cutting require more oxygen consumption than other tasks. We next scaled these oxygen consumption rates to the individual brain mass of the workers in order to reveal mass-specific metabolic rates. The red line here is the Adacephalotes species I've been talking about, and the black line represents monomorphic species. Monomorphic species are much less socially complex because they live in smaller colonies and all the workers are roughly the same size as shown in the picture in the upper right corner, and all of the workers participate in all of the tasks rather than dividing labor roles among the colony. So what we see when we do this is that the smallest ADA minim workers actually have more efficient brains than similarly sized monomorphic workers, which reveals that task specialization likely leads to the efficiency of brain metabolism. However, when we look at the medium-sized workers, they do not show decreased oxygen consumption rates for their brain mass compared to the monomorphic species, which may reflect the fact that we think these media subcastes are more so task generalists and would therefore have similar sized task repertoires to the monomorphic generalist workers. So in conclusion, my results across these 3 collateral studies suggest that something interesting is going on with the medium-sized workers in ants. They may be task generalists as they have higher investment in these higher order processing regions, and this conclusion is further supported by their unique gene expression profiles, as well as the large variation seen in their individual brain oxygen consumption rates. I would like to thank my PI, James Traniello, for his support and guidance over these past few years. And I would like to especially thank my 2 graduate student advisors, Zach and Isabella, without whom this project would have been completely impossible. Thank you.